All right. Hi, everybody. This is the recording or the review recording for our first exam. It's just basically an audible narration for those that are not familiar with how I do it, though most of you are. I just basically peruse the test. I take a look at the exam as I have it set up and get on here. And I'm just looking at the exam. Again, this is Blackboard, and I'll start commenting. Interesting thing with the Blackboard exam, again, is the time. I urge you to be prepared ahead of time. I think there's 45 questions, it turns out, and I would dare say it's 15 to 30, like 15 on chapter one, which we've completed, and 30 on the chapter we're going to be doing the most of. At least it'll be the most recent. So you need to start studying for the stuff that have presented already. Started on Friday in earnest. Monday's a big day. This is Sunday the 2nd, depending on when you're listening to it. So the lecture from the 3rd, which will have gram negatives and gram positives, extraordinarily important. And we'll probably get through all this stuff Friday and maybe just a little patch for Monday, but it should be good. All right. So historical figures are part of it. Pasteur, remember what we're talking about. It's all between something they called abiogenesis versus, which was spontaneous generation, life out of lifelessness. It's something would magically come out of nowhere and start to grow. All of a sudden you get a disease like an act of God or the devil or something that they did not understand, some evil force or unseen force in the air, as opposed to particles that couldn't be seen, which is germ theory. And that's really what a lot of those historical figures, starting with Reddy and the meat and the flies and the glasses, and then realizing that it was worms laying eggs that led to maggots, not like some force magically created a life out of an old piece of meat that was sitting out, or basically the other things that were done. Semmelweis and his ideas about cleanliness, particularly associated with puerperal sepsis. They didn't know it was streptococcus, but he did. It was similar to infection that he'd seen someone die of, so he reasoned that they were carrying whatever this infection came with, whether it was coming from the lab, and they weren't being careful washing their hands, and he instituted all these procedures, even though the medical community was very angry at him. He was absolutely right. And then Lister around the same time as Pasteur, surgical instruments and cleansing them and cleansing the skin and cleansing their hands, much more on top of, similar to what we do today. Those are the aseptic principles. That's why we consider Lister being the father of what we call modern aseptic technique. Sterile technique is really a somewhat older term for that, though it's still used a lot. And then, of course, Pasteur. And Pasteur really, had a very understandable scientific experiment with this gooseneck flask. Problem was the broth. Talked about it in class. His premise was boiling killed it, but because that worked, as long as you didn't have endospores. And he was not aware that in the soil, when they used hay infusions, which are very common for a lot of purposes in and around, and particularly so that people had at homes then, using remnants of things that weren't sold for broths and things like that and food sources, how that potentially could create contamination. And that, I understand, the endospores were not destroyed by that. And eventually that was proven as they identified the endospores, and Koch actually used an endospore, anthrax. Uh, he hadn't named the disease until the disease was called anthrax, but he began to identify that was, even though he didn't know it was an endospore former, it did show up microscopically. But that was the one with Koch's postulates, establishing the chain of evidence, as I described it. And of course, he was the lab guy who identified all the many bacteria that we'll be looking at the lab continuously, starting in the labs this week as we start to gram stain. He grew them in pure culture. That was his way of identifying them. That was his way of proving things. Where Pasteur was much more about treatments. He coined the term virus, which meant poison. He reasoned there was something that he couldn't even see with his microscopes that he had that was also a particle of some type, unseen in the air. But he found ways to treat it. I mean, here he was with this rabies, which was a death sentence if you were bitten by a rabid animal. Yet he began to take fluid from animals that seemed to be improving, even though they had the rabies, and realized that there were probably something in their blood that he could take out. And effectively, what they do is they use different kinds of serum. Typically, horse serum was used for a lot of things, and the serum basically you don't have to know this stuff. But they spin the blood down, and the serum sits on top and suck it off. We talk about this in AP2, and probably refine that in some way, shape, or form. And that was one of the first treatment for rabies. So then Pasteur and his institute were all about treatments. I think that's a big deal. So Pasteur was all about proving germ theory as opposed to spontaneous generation. And so, but when we talk about the first vaccine, okay, we're really talking about Jenner and smallpox, and he preceded all of these people because he was before 1800, where Pasteur was around the time on or shortly after our Civil War. And so you can remember all of those. And yeah, and Koch, if you think of a bacteria, tuberculosis, leprosy, staphylococcus, streptococcus, anthrax, E. coli, he identified them all in the lab. They developed augers, they developed pure culture techniques that many of you will be using in lab this week. My lab already started with it, etc. So there's just so much to know about why they're so valuable. Genetic engineering, we typically utilize simple organisms like E. coli and yeast because they're easy to manipulate. Simple yeasts are very versatile. They're such a simple eukaryotic cell that we can splice things into them. A lot of research is done on yeast as it is on bacteria because, frankly, all these microorganisms, they kind of do business the way we do business. And in that sense, I mean, they ingest organic chemicals, they do energy transformations, they utilize proteins and carbohydrates and fats, and they either extract energy, they use them for building blocks, just like we do. And so things that will help or hurt our system, we're able to see that. But in genetic engineering, we typically alter their DNA. There's RNA technique, but the big deal is DNA. That's true genetic engineering. We see genetically modified organisms like GMO, or making insulin, the first one of these, or making human growth hormone, or making hepatitis B immunizations. That's DNA, and it's in E. coli and yeast is where we find most of it. Emergent and resurgent is an important concept. Emergent diseases are just that. And probably due to mutation, probably going into areas we haven't been to. Better techniques in identifying them. So, effectively, COVID-19, whether it came from a lab or came from a market, was still a mutation of certain kinds of viruses, which we'll learn about. Of RNA viruses can flip-flop genetic sequences between each other in different species, and sometimes you come up with a real bad oddball. That was COVID-19. MRSA, which is another example, is a mutation in that developed because of exposure to antibiotics. So it's actually a defense mechanism, but it emerges, and we've actually evolved that, as we'll talk about Staphylococcus a couple of times since we discovered antibiotics. Where things like tuberculosis and measles are resurgent, you remember the reasons. Vaccine laxity would be measles, worldwide travel, immunosuppressive drugs, immunosuppressive diseases like HIV, aging populations, all of these, a lot of diseases that we thought were eradicated to sort of rear their ugly head. Again, remember Semmelweis and Lister, the roles they had, Pasteur and Koch, there's questions on them. We talked about basic bacteria 101, the big deal, peptidoglycan. It's the only place we find it. Why it's such a good target and why penicillin's all about it. So you need to remember that. And then we have different, there really are subkingdoms of eukaryotic microbes. Those, we call them protists. And really, what algae is the one with chlorophyll, that makes it unusual. It's in the water, that's not so unusual. We have the land-based one that's also in the water is protozoans. Those are those single-celled organisms where algae are single or multicellular, but the chlorophyll sets them apart. Algae are more like plants because they have cell walls. Protozoans are not. They're more like individual animal cells, like our kind of a cell. And we look at them, and they're very important for diseases. I told you 16 times already that the second most common sexually transmitted disease, Trichomonas vaginalis, is from a protozoan. Think about that. Everybody hears about HPV and herpes and HIV. So, I mean, you're talking viruses there. And syphilis is a bacteria, and gonorrhea is a bacteria. This is protozoan, so you can't ignore those. And they are very significant and a part of this course as well. And then sort of lastly, when you get fungi and mold and yeast, they do not have chlorophyll. They are principally land-based, and for the most part, we call them they basically saprobes. SAPBROBS, I think it is SAPROBES. Pardon me, and they're decomposers. They break stuff down, all the bacteria, kind of like roadkill deteriorating. And what do they have? Again, they have a cell wall, but they don't have chlorophyll. So they're almost like a chlorophyll-less plant, if that makes any sense. It's one way you want to remember it. And remember the important things like fixing carbon and fixing nitrogen. That's fixing carbon is how we get carbohydrates. Plants give this, they give us air. We saw how important they are for creating oxygen in the environment, algae in the water. Fixing carbon means taking carbon dioxide and affixing, not repairing, but stapling it together. Same thing with nitrogen, taking nitrogen and stapling that on carbon-containing organic compounds, and it's the basis for proteins, and it's the basis for nucleotides because they are nitrogenated, if you will. The distinction between bacteria and archaea is really the environments they are and their cell wall, because bacteria have peptidoglycan, archaea don't. Archaea live in extreme environments. They're very similar in size otherwise that are there. Viruses, we're going to watch out for them. They can infect everything. Every living thing can be infected by a virus. E. coli gets a virus. You get a virus. I get a virus. And remember, DNA or RNA, never both. The distinction between them and prions. Prions are similar. They're non-living. They're not made out of cells like viruses, but they're just proteins. They involve no nucleic acids. I think that's important. We haven't talked about endospores at length yet, but we will. There's wonderful animation. And what they do is they're typically found for our purposes in only two genuses. We call Bacillus species and Clostridia species. Clostridia, we use the Bacillus in the lab because it's aerobic. Clostridia, as I said, was anaerobic. So that's something that's important. And understand we're going to see a whole animation about endospores are a state. Bacteria are like, we thought they were plants. So we say if they're in a vegetative state. Students get this wrong constantly. You think vegetative state, you see somebody who's in a coma, somebody who's irresponsive at end stage Alzheimer's or end stage other kinds of mental diseases, or they've been injured severely and there's nothing happening. We're just kind of waiting for them to expire. For us, that's a vegetative state. For plants and plant-like structures, which we thought bacteria are, vegetating means they're happy, they're growing, they're multiplying. So endospores go from the vegetative state, when they're basically a productive bacteria, to not doing anything. So it's kind of interesting. Just waving my wife goodbye. She's going to go out shopping. So they are in this interesting state. They can last forever, hundreds and hundreds of years. It's an asymmetric division of a particular bacteria. The smaller part is good, and they're the same genetically, but the cytoplasm and surrounding material is much less. It's called a forespore, for means before a full spore, and remnants are called a mother cell. You'll see that. And what happens is that eventually it forms layers of peptidoglycan. You'll see this in between two membranes. You'll see this clearly in the animation. And basically, eventually, proteins come from the mother cell, from the remaining enclosure, and create an additional coat. That's why they live so long. They have very little. It's almost like they're in a state of suspended animation. It's almost like if you put something in a food dehydrator. Or it's almost like creating a little seed that you just, what makes it form if there's not enough nutrition. Bacteria that form endospores form them when there's not enough carbon and not enough nitrogen or not enough water. And what happens is when all those essential elements are restored and the conditions are favorable, they start to grow again, which means they go from the endospore stage to the vegetative stage. It is not a type of reproduction. I will show you this at length. If you have not seen the animation, we'll talk about this in class. Remember, the forms cocci are more spherical. They look like circles. Bacilli look oblong or rectangular. They're little rods, but there's variations. You all know what cell membranes are. Phospholipid bilayer, embedded protein, semi-permeable is sort of the definition for us. We all know about diffusion and osmosis and facilitated diffusion. We're reviewing them. The big driving force is always concentration gradient. Remember, they do not have membrane-bound organelles. They have organelles, but they're not membrane-bound. Membrane-bound means they're in a container, like the nucleus, the endoplasmic reticulum, lysosomes, Golgi apparatus. Okay. Ribosomes are little dots where they make proteins, tiny little areas, and only in eukaryotic cells do they ever get bound with membrane. There's also free ribosomes. Bacteria have free ribosomes. They're in the bacterial cytoplasm. They are 70S by nature. They all do protein synthesis. And a ribosome is ribosomal RNA. That's where the 70S comes from, and proteins. We're going to go over peptidoglycan. We know it's only found in bacteria. But gram positives have a lot, gram negatives have comparatively little. That's the big difference. You're going to see the composition with these unique nitrogenous carbohydrates. We're going to name NAMs and NAGs, how they alternate, how they're sort of stapled together with tetrapeptide groups and peptide interbridges. But we're going to have to, I'm going to remind you, if you're not hearing, if I'm doing something like that, tetrapeptides and interbridges, I'm doing it in passing. I'll accentuate what's important, like NAMs and NAGs. You don't even have to know the whole name. Gram negatives, very unique because they have a very little bit of peptidoglycan. Basically, from outside to inside, you have an outer membrane that's very unique. We'll go over it in a second. You have an inner membrane that's normal. In between, you have a thin layer or two of peptidoglycan rather than 30 to 40 layers in a gram-positive cell on the outside. And you have an interesting kind of cytoplasm we call periplasm. It's not true cytoplasm because it's not inside the cell and not within the cell membrane, but it's still within the cell. It's sort of a composite that's in this interesting cellular envelope. So the lipopolysaccharide layer is part of what is the unique thing about the gram-negative cell wall. It's not a phospholipid bilayer. It's got phospholipid on the interior, you will see, but it has a lipid polysaccharide layer, which is different. The interior lipid portion, the exterior lipid portion is inherently toxic to us. But as long as the bacteria are contained someplace where they're not exposed to sterile tissue, or we're not having extensive amounts that our immune system can't take care of, they don't bother us. But if you burst an appendix, all of a sudden all of these gram-negative bacteria that are in your gut pour into your abdominal peritoneal cavity, you get a thing called peritonitis, you can die, and quickly. That's called gram-negative sepsis because it's inherently toxic to us. We have gram-negatives in our respiratory system, in our mouth, all the time, but those are areas that are by no means sterile. Okay? It's when they get inside that they can do damage. And we'll talk about this in the way infections go as well. So you're going to see something called the O-specific antigen or polysaccharide that extends from this lipid A component externally, and that's an identifier. So when we're classifying, well, we have E. coli, but not every E. coli can hurt us. The way we identify which E. coli can hurt us, we use these identifying antigens that are found on this O polysaccharide and on the flagella, interestingly enough, and we'll identify those down the road as well. By definition, since it's a toxin that's part of it, gram-negative, we call it an endotoxin. Only gram-negatives have that, and it's only the lipid A portion, where all bacteria make exotoxins. That's a bad statement. All bacteria, both gram-negative and gram-positive, have the capability of producing exotoxins, toxins that they give off that can hurt you. and frequently do. So usually when you get sick from a bacterial infection, it's the toxins. Sometimes it's just laying there. Most of the time it's the toxins. And again, when you think about these nasty strains of E. coli, they release exotoxins, and they have a lipid A thing, so they are inherently an endotoxin. Staphylococcus and streptococcus are gram-positive. They do most of their damage by releasing toxins or creating an immunological response to it. So that's all important. The other thing about gram negatives, that outer membrane makes them impervious a lot to water soluble things. Some water can get through because there's little water pores or porin, P-O-R-I-N proteins in the membrane, as there are in all membranes, just about both inner and outer membranes. How does water get into a gram-positive cell? A gram-positive cell is kind of like, as you'll see, kind of like a regular bacterial cell that has a cell membrane, but it's got a cell wall. It's basically just 30, 40 layers of peptidoglycan. Peptidoglycan is porous inherently. It's not like it took a great big piece of saran wrap and wrapped it up. Okay? That wouldn't let water in. It is inherently porous. That's there. So it's think of a material that you can put water on the outside, and eventually it'll drizzle its way through the inside. That's what porosity is. All of these have sugars that are attached to proteins on the exterior, just like all our cells do. It's called glycocalyx, a sugar shell. They can aggregate. They're basically polysaccharides, which are complex sugars that protrude, and they aggregate. And if they're very thick, they can make they can make capsules, which are really important in respiratory diseases like tuberculosis, like certain kinds of streptococcal infections, streptococcal pneumonia. And sometimes they're just sort of in disarray. And we talked about it a little bit. They make what we call a slime layer. Slime layers are really being investigated now. We use them to treat water spills, but your dental tartar is a slime layer that accounts to a fairly significant extent for why we get cavities. We talked a little bit about flagella already. They're made of flagella, and these are bacterial flagella. Remember, a lot of these questions are in prokarya or eukarya. Prokaryotic cells have flagellin. Eukaryotic cells, the microtubules, are made of that 9+2 pattern that we went over in AP1. We're going to go over it again because eukaryotic cells are microbes also. We'll talk about the fertility pilus. I touched it in passing in most of the lecture I did the Friday before I'm doing this dictation, which would have been the 31st of January. That's a hollow protein made out of something called pilin, not microtubules, and it's useful for DNA transfer. That's how genetic material goes from one bacteria to the other in the same generation. And that's really going to be an important point. That's a focal point, as are plasmids, which are the things that are transferred usually by those pili, is the appropriate plural. And that's just a little circle of DNA, good old double-stranded DNA, by definition with useful but non-essential information. What it means is it typically has resistance to antibiotics. It has ways to survive in its environment, but the bacteria can survive without it. That's what means non-essential. It doesn't have to have it to live. The definition scientifically of essentiality is that it needs it to survive. Like we have to have air and water and food to survive. If we don't have it, we are going to die. Bacteria are helped by plasmids. They don't need them. And a lot of times they disappear when they're no longer needed. Remember, 70S ribosomes in bacteria, 80S in eukarya. The problem with us in eukaryotic cells with 70S ribosomes, we have them in mitochondria. And that's why antibiotics that target ribosomes, which many of them do, even the Z-Pak and really common ones like tetracycline, okay, and tetracycline derivatives target the mitochondria. They cause side effects where antibiotics target peptidoglycan, like penicillins and their cousins cephalosporins, have more problems with allergies. We go over this in antibiotics, but it's something that's really the rationale or behind it. And other things to note, again, with the endospores, I forgot to mention that it really takes several hours for them to form. Once they start to form, they form continuously, as long as conditions like that. But remember, it's about not enough basically food or water like nitrogen and carbon containing sources. That's what we live on too. Okay. Remember, different things, cells get stability from having a cell wall in bacteria. I remember I mentioned that the one called mycoplasma utilizes sterols, Sterol. That's something that's derived from cholesterol. Cholesterol is what we use in animal eukaryotic cells. We don't have a cell wall. Ergosterol, Ergosterol, is used in fungal cells, even though they have a cell wall. And actually, is a target we utilize when we do antifungals. So, I mean, bacteria generally don't have sterols with the exception of mycoplasma and maybe a few others. But it's all about cell membrane stability. There isn't much of a cytoskeleton. They have tiny mints of little intermediate filaments. It's more than with these almost nanofilaments. So we're just starting to realize them now. Eukaryotic cells like ours have intermediate filaments and microtubules. Of course, we have all the other stuff in eukaryotic cells: nucleus, nucleolus, Golgi apparatus, lysosome, peroxisome, da 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 da. So that's all part of it as well. So you remember the anatomy from outside to inside or inside to outside of the gram-negative. Remember, outer membrane, periplasm with the peptidoglycan, inner membrane, which is the cell membrane, then into the cytoplasm. Okay, and peptidoglycan we're going to get into in length. Again, it's porosity. One of the ways it attaches to the bacterial cell membrane is called teichoic acids. It's one of the things we stain for in the lab when we use basic staining. When we're going to talk about that, I will, in staining this week for gram stains, and the fact that these are the NAMs and NAGs are made out of nitrogenated carbohydrates. That's not unusual. Our connective tissue, that gel like material that we have in our connective tissue. Remember, the connective tissue has cells, fibers, and ground substance. The ground substance in our connective tissue, nitrogenated carbohydrate. You might remember the term glycans, Glycans. We covered run, tumble, run, all the things they're attracted to or repelled by. Remember, heat, light, magnetism, all of those types of things. So chemicals, magnetic particles, light, air, temperature, all things that can either attract or repel: chemotaxis, thermotaxis, da, 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 da. You know about the organelles like mitochondria and chloroplasts because they're part of microbial cells as well, but eukaryotic, that's where we do energy transformations. We don't do protein synthesis there. We're going to see bacteria are unique because they don't do much. They're asexual. All they do is binary fission. And that's where they beat us. They keep dividing in two. We talked about that extensively in physiology, but it's a defining factor. And the difference: phospholipid bilayers are everywhere. Don't be surprised about them. That's a standard living membrane. So bacteria have it and we have it. Again, that outer membrane of the gram-negative is an oddball. Let's see, reinforcing plasmids. Most bacteria have them, and they typically have more than one, and more than one can move at the same time. You'll find out. The big distinction about viruses: DNA or RNA, never both. Don't get that wrong. I'd be very upset about things like that. How the DNA is arranged, I forgot to mention that. DNA, we have eukaryotic cells, it's pairs, linear pairs of double-stranded DNA. Bacteria have a circle of DNA that happens to be supercoiled. It's all ready to reproduce. Same double-stranded DNA in both, but different configurations, and that's important. And I think that's everything. So thanks a lot. I'm sure you'll have questions. I'll respond to emails. I'll distribute this to you when it is available. And I believe I'm good to end this. Bye-bye.